My name is Emily Ackerman. I'm a grad student at the University of Pittsburgh, and today I'll be talking about how we've identified regulators of viral infection using virus host networks. Every year, there are seasonal outbreaks of virus infection. Here I'm showing you the 2009 swine flu, as well as the current COVID-19 pandemic. And when we have these outbreaks, we need therapeutic treatments quickly. But the problem is, that's a difficult thing to do. For example, for the flu, there are only 4 FDA-approved treatments, 2 of which are not suitable for people who need them the most. And for COVID-19, as we all know, there are no antiviral treatments available. So the question is, can we use existing PPI data to efficiently predict drug targets and candidates in a new way? And to do that, we've employed virus host protein networks. Here, for example, we have a network where there's this large group of proteins that don't interact with the virus, and a smaller group that do. Here, for example, is influenza A. Traditionally, we've used topology to look at those virus-interacting proteins and understand that they're significant to the network and therefore to disease. But networks are dynamic, they're highly connected, and so we wanted to move beyond topology to understand how these networks are affecting a viral infection. So we've developed 2 methods of understanding these regulators of disease for influenza A and SARS-CoV-2. The first method we've applied to influenza A. It's a virus-specific subnetwork extraction where we take data about virus-interacting host proteins, shown here in blue, and proteins that, using siRNA data, we've identified as important to viral replication, here in orange, and we extract all the paths between them and call those, in gray, the connecting proteins. And this subnetwork is representative of the disease events that happen after infection. And so we use this subnetwork to identify potential drug targets or to understand if this network is enriched for potential drug targets. So we define this hit rate of enrichment, and we look at all proteins which have a very low hit rate, maybe 0.02. When we look at this traditional group of virus-interacting proteins having to do with transcription, translation, we see the hit rate is about 0.23. But our new group of proteins, the connecting proteins, turns out that they have to do more with the immune response, particularly the NF-kappa B pathway, and their hit rate is even higher, 0.35. And when we look at topology of those proteins here, the high and low between this are segmented, there isn't much of a difference. So what's important about this subnetwork is that we're identifying a group of proteins that's traditionally not been identified by network studies, yet has great potential for being drug target candidates. Now moving to approach 2, we're motivated to push the PPI further from a static topological snapshot to a dynamic system. And so the question becomes, how do viruses control the cell's biological pathways? So we take a healthy host PPI and an infected virus host PPI and use this controllability analysis, kind of a black box right now, or a pink box, but it's 2 analyses where the output are key regulators of the infected cell based on the differences in regulation of these 2 networks. And from there, we can identify the topology, the functional roles, as well as any potential for drug repurposing of these regulators. So we've applied this to influenza A, which has been published, and SARS-CoV-2, which is under review currently. Both have identified a small subset of proteins with low overlap. I'm just going to carry on with SARS-CoV-2 as an example. if we look at the topology of these proteins, we see that there's this really interesting group, for both viruses actually, of proteins where in the healthy cell, the HIN here, there is a betweenness of 0. But in the infected cell, they have high betweenness, meaning that their significance to the network flow of this cellular state is unique to the infected cell. And when we go into the biological roles of these proteins, we find that they're interferon regulated, meaning that once the interferon response kicks in, they see a fold change up or down regulation of 2 in these genes, meaning we're identifying proteins computationally that are both topologically and biologically relevant to infection. And further, if we look at these proteins, 8 of them are already known to have a compound that targets them as a drug target. 6 of our identified proteins were also identified by Gordon et al. In this kind of keystone paper so far. And we get some really interesting targets. So we have the hydroxychloroquine target having to do with cell stress. We have RNA synthesis, a known immunosuppressant target. We have genes having to do with nuclear transport and translation regulation. So all of these are important for a viral infection. And having identified them computationally and having them align with experimental results is a really great validation for this kind of method. Even further, 3 of them have known compounds that experimentally inhibit viral replication of SARS-CoV-2 specifically. So we're already on a great start towards repurposing drugs for SARS-CoV-2. Additionally, there's 2 targets that we've identified that have not been identified in the context of COVID-19. So these are under development by us, and they will be out soon, so keep your eyes open for them. In summary, we've used these virus host networks to reveal disease-specific regulators of infection. And by doing this, we've found traditionally unidentified protein groups and prioritized drug repurposing efforts using existing data in a very short period of time. So I'd like to thank my group and our collaborators as well as my funding sources. I'm happy to talk to anyone about this work. So thank you for listening.